Okay, so let's start, guys. So, good afternoon, everyone. Today we'll be starting with the discussion of week 5 in the course Experimental Biotechnology. So, we'll be discussing about the gel filtration chromatography in this lecture. Okay, so if you guys have any doubt, please stop me on any slide if you want. Okay, so let's start. So the first question is gel filtration chromatography separates proteins on the basis of their options are charge, size, hydrophobicity or affinity. Yes. So the correct answer is size. So in case of gel filtration chromatography, what we have is the basic structure. This is the column. And here in this column, we fit in beads that have a specific pore size. So like this, we fit in beads, and in these beads, we have a specific pores, like we saw in electrophoresis. Okay, so in this case, we have pores, and based upon the size of the particles that we have to separate, the there would be different sizes, and based upon the size the particles will enter the pore and will travel as much distance as possible as compared to their size, and the particles that have a size greater than the pore size will travel outside the beads and will elute out first in case of the gel filtration chromatography, and the particles that have size in range of the pore size, those will travel through the pores and it will take longer time to elute out, and they will elute out on the basis of their size. Okay, so the correct answer is option number 2. Charge is used in ion exchange chromatography and as well as chromatofocusing. So chromatofocusing is like isoelectric focusing, but here we use the pi as their separation component, and then hydrophobicity is used in hydrophobic interaction chromatography, and affinity is used in affinity chromatography. Okay, so any doubt in this question? Okay, if not, then we'll move on to the next question. So the total volume of a gel is equal to the sum of pore volume, void volume, elution volume, pore volume, protein injection volume or void volume, protein injection volume, void volume or elution volume, and pore volume, void volume, protein injection volume. What can be the correct answer? Okay, someone said first. Any more answers? Okay, someone said second also. Any more? Okay, so in this case, what will happen? For example, this will be the column. Okay, here we have the beads, that have specific pore size packed in a column. So the particles will follow that are in range of the size, so that will follow through these pores, and that, they will flow out. So the pore volume is the volume that is occupied inside these beads, okay, where the small particles that are being separated in the chromatography will travel. So here this will be the pore volume. Okay, let's depict it with pi, and the volume that is outside these beads, that is the a void volume. Okay, because it is not serving any purpose, it's just the flow of the large particles that cannot enter the beads. So that is why it's the void volume. Let's depict it by v naught. Okay, now comes the elution volume. So in case of the elution volume, what will happen? The volume that will travel through both pores as well as the void volume. Okay, so it will be constituent of all the things that are there except the gel. Okay, so that will constitute the elution volume, that we'll collect it afterwards to detect the number of protein and the how the proteins are separated through the gel filtration volume. So now what is the question? We have the like, and there is another term that is protein injection volume. So the sample that we load, okay, that will be the protein injection volume. Okay, let's depict it by vt. So now we have to calculate the total volume of the gel. Okay, not the total volume of the column. We have to calculate the volume of the gel. So in that case, that will be equal to the pore volume, okay, the void volume, and the elution volume, because all these three will constitute the area or the volume that have constituted by the gel. Okay, so this will be the correct answer. So the correct answer would be option number one. Okay, so any doubts in this question? So if not, then we'll move on to the next question. Given below are the Kd values of different proteins. So Kd is the distribution coefficient. Okay, so we have four proteins and the Kd values are as follows. For protein A, it's zero point 0.5, B, it's zero point 0.8, C, it's zero point 0.3, and D, it's 1. So what is the order of elution in this case? Okay, someone said B. Okay, any more answers? Okay, just one minute. Okay, so what happens here is, to remember, Kd is inversely proportional to molecular weight. Okay, so whenever molecular weight will be high, the Kd will be less, and the less the Kd, the more quickly molecules will flow out or elute out. That means the retention time would be less. Okay, so here if we have the least KD, that means that will elute out first in that case, and that will have the most or the highest molecular weight in that particular set of proteins. So here the KD of protein C is the least. Okay, so in that case it will elute out first. So the first would be protein C, then comes zero point 0.5, that is protein A, then zero point 0.8, that is protein B, and then comes protein D. Okay, so this is the correct order, and this is depicted in option number 3. So yes, this is the correct answer. Okay, so any doubts in this question, anybody? Okay, if not, then we'll move on to the next question. The distribution coefficient Kd in gel filtration chromatography is independent of the void volume, elution volume, column length, or pore volume. What can be the correct answer? Okay, someone said fourth. Other answers? Okay, any more answers? Okay, so what is the formula for Kd? Can anybody tell? How do we calculate Kd? In the last class we discussed. So it is concentration of the analyte uh, in phase A and divided by the concentration of analyte in phase B. Okay, so here in this particular case, the column or the characteristics of the column, okay, will not depend or will not affect the Kd value, because it is dependent upon the only the concentration of the analyte in the either of the phases. Okay, either in the stationary phase or in the mobile phase. So in that case it will not be affected by the column characteristics like column length or column height. Okay, so here the void volume, elution volume, and pore volume are basically characteristics that are related to the analyte, because in that case what will happen, the elution volume, void volume, and pore volume are characteristics that are dependent upon the analyte and its molecular weight. That's how we calculate the molecular weight and the elution volume, retention time of the analyte in the gel filtration column. Okay, so now the thing that is left is column length. So column length is independent of the distribution coefficient of the analyte in the gel filtration chromatography. Okay, so any doubts in this question? So basically, the KD KD definition is like it's a ratio of concentration of the analyte in phase A, that can be the stationary or the mobile phase, and divided by the concentration of the analyte in phase B. Okay, so here KD is basically characteristics of the analyte rather than the column. Okay, so in that case, if we consider a column that have a particular beads in it with a pore volume and a void volume. So here the volumes that we use are basically used to determine what will be the like separation of the protein, how it will take place. So in that case, the volumes are used for the analyte to for our purpose to how the analyte is separated. Okay, so in that case, the column length would not affect the KD of particular analyte. So that's why the column length is basically independent, or it does not affect the distribution coefficient of the uh, analyte. Am I clear? Okay. So, any more doubts? If not, then we'll move on to the next question. So, if distribution coefficient is greater than 1, then the options are: protein is eluted first from the column, protein is absorbed on the beads and will not elute, protein is eluted completely from the column, or protein will enter completely into the pore and elute last. What can be the correct answer? Okay, any more answers? Okay, so yes, the correct answer is option number 2. So what happens in this case? The formula that we use for calculating KD is, so this is the pore volume, this is the void volume, and this is the elution volume. So in that case, if we have KD that is greater than 1, so in this means that uh, sorry, VE will be less than. So in that case, yeah. So in that case, what will happen? The analyte will go and get absorbed on the beads and will not elute out. Okay. So in that case, the KD will be greater than 1. Okay. So any doubts in this question? Okay. If not, then we'll move on to the next question. Choice of column in gel filtration chromatography depends upon the charge and mass of the protein, pressure limit of the equipment and charge of protein, pressure limit of the equipment, or pressure limit of the equipment and the size of the protein. What can be the correct answer? Okay. So yes, the correct answer is option number 4, because here we have to check for one thing. First is the pressure that we are putting on the equipment. Okay, because the amount it will handle and it can handle to separate or segregate out, This is a major component. And second, the most important, on the basis of which the separation would be possible, was the size of the protein. Okay, so the correct answer is option number 4. Okay, so any doubts in this question? If not, then we'll move on to the next question. Which matrix is used for protein of 300 kilodalton to be separated out? Cepadex G25, 4100, G10, or G50? Okay. Any more answers? So the correct answer is option number 3. Okay. So this is the list of the proteins, the columns that we use generally for uh, separating different proteins. Okay. So, for example, Cepatex G10 is can separate up to proteins that are 700 daltons in size. Okay, G25 can separate between 1000 and 5000 daltons. So this is a particular range, and based upon that, we have the name of the particular bead, and that follows. Okay, so these are different Cepatex columns, and here is the molecular weight that is mentioned for this. So the highest is Cepharo 6b, that can separate between 10,000 to 40 lakhs. Okay, daltons. So this is the this will be the highest, Cephalus 4b, and these two are basically used for separating large molecular weight proteins. Okay, so here the protein that we have to separate is 300 kilodalton. So it basically ranges in this G200. Okay, but it is not in the option. So this might be 300 daltons. That can be the correction that can be made. So in that case, G10 can be used to separate these particular proteins. Okay, so this questions was a bit incorrect, because here the 300 Dalton would be the correct question. Okay, so these are different Cepadex columns that are available commercially, and based upon our need, that the protein that we need to be separated, basically we use the different Cepadex for those particular proteins. Okay, so this is the case with the Cepadex columns. So let's move on to the next question. Which of the following follows a linear relationship in gel filtration chromatography? Elution volume and log of Kd, log of elution time and Kd, log of molecular weight and Kd, or elution volume and Kd? Okay, anybody else? So the correct answer is option number 3, and here is the explanation. So whenever we separate proteins on in a gel filtration column, so here is on the y-axis we have the KAV or the KD, okay, and on the x-axis we have log of molecular weight. So based upon this, as we said, although it's a linear graph, but it's an inversely proportional. How? If the KD is less, here the molecular weight is high. Okay, and as we move forward, as the KD increases, the molecular weight decreases. Okay, so it's a inverse relationship because of the log. If we plot it as a KD and molecular weight, so in this case the graph would be like this. Okay, because it follows a inverse relationship. Okay, so is this question clear? Okay, so let's move on to the next question. Dash is used to calculate void volume of freshly packed Cepadex 75 column. Ethanol, blue dextran, methylene blue, or acetone. Okay, any more answers? Okay, so what is void volume first? The volume that is outside the beads. Okay, so in case we have to calculate the void volume, so the protein that we are using or any control that we are using to calculate the void volume must not enter the beads. Okay, this is prohibited. Then only we'll be able to calculate the volume of the voids or the void volume. Okay, so in that case we will not use acetone because acetone is a small molecule. Okay, and it is a general solvent that we use to dissolve various proteins that are being separated in a column. Okay, so if we use acetone, then it will flow both in the void volume as well as the pore volume. So we'll not be able to detect the amount of the like what will be the void volume. So in that case, in void for calculating the void volume, we'll use blue dextran. Okay, so blue dextran have a molecular weight of nearly 2000 kilodalton. Okay, so what will happen if we use blue dextran? So in that case, it will flow only through the voids and it will not be able to enter the beads or the pores. So because its size is greater than the pore volume that we'll use in Cepadex 75 column. So it will flow through the voids and it will calculate the total void volume. Okay, so the correct answer is option number 2. So okay, so moving on to the next question. Which of the following is used to calculate the total volume of the gel filtration column? Okay, now I think you guys can answer this question. Yes, in this case, it will be acetone because acetone is a very small molecule and it is generally used as a solvent for dissolution of mostly all the proteins that we use to separate in a gel filtration column. So it will flow through both the pores. So it will flow through both the pores as well as the voids. Okay, so it can. be used to calculate the total volume of the gel filtration column. Okay, so any doubts in this question? Okay, moving on to the next question. Which of the following is done to remove precipitated protein from the gel filtration column? Washing the column with 20% ethanol, washing the column with buffer, flowing small volume of protease through the column, or reverse washing column with water. What can be done in this case? Okay, any more answers? So yes, sometimes it happens whenever we are separating any proteins through the column because of the various reasons, the pH of the buffer or any other reason, the protein can get precipitated inside the column itself. Okay, so it will go and it will attach or it will be stuck inside the beads. So in that case, to remove those beads or to remove those proteins, what we flow the column with small amount of protease. So what will protease do? It will degrade all the proteins that are present or precipitated inside the column, and that will flow out as a small chunks of proteins. Okay, small fragments of proteins. So this is the correct answer, option number 3. Okay, so any doubts in this question? If not, then we'll move on to the next question. Which of the following experimental setup is suitable for determining the oligomeric state of the protein? Affinity chromatography followed by native page, gel filtration chromatography followed by native page, gel filtration chromatography followed by SDS page, or gel filtration chromatography of denatured protein followed by page. Okay, so yes, the correct answer is option number 3. Okay, so in option number 1, we have the affinity chromatography followed by the native page. It will not be able to detect the oligomeric state because in case of native page, it is the whole protein. Okay, so for example, if a protein has two subunits, so in this case, it will appear as a whole protein with the whole molecular mass. And affinity chromatography will not be able to separate on the basis of the size. Okay, so this option is neglected. In case of second option, both are following the same function itself. So in case of the oligomeric state, it would have more molecular weight as compared to the single state. Okay, so in case of gel filtration chromatography, it will be eluted first, and in case of native page also, it will be eluted as a whole, as a dimeric state. Okay, so this will not be able to serve the purpose that we are using. So in the third case, gel filtration chromatography followed by SDS page can be used. So what will happen, if we have the oligomeric state, we can separate it through the gel filtration column, followed by which we can use the denaturation process, like using beta me or beta mercaptoethanol and SDS. So this will denature the protein, and on that basis we'll be able to get the monomeric proteins or monomeric subunits. Okay, so this is the correct answer. And the third option is gel filtration chromatography of the denatured protein followed by page. So in this case also we will not be able to detect the oligomeric state because in the first step itself, it will denature the protein. So here it will not be a dimeric form of the protein, it will be the monomeric form, and that only will run on a phase gel. So we'll not be able to detect whether it's a dimeric or a multimeric protein. So this will only be the monomeric form of the protein. Okay. So any doubts in this question? Okay. If not, then we'll move on to the next question. Protein X from Mycobacterium tuberculosis strain is treated with 3 molar urea. What will be the resultant effect of the gel filtration chromatography of urea treated protein X with respect to the untreated protein X? And these are the options. So, what can be the correct answer? Okay, any more answers? So, in this particular case, we have to understand one thing. I have taught that thing in the last lecture also. So, if we are having a fully folded state of a protein, okay, so it's the scientific term for that is hydrodynamic area of this particular protein would be less. Okay, if we treat this protein with urea, so urea is a denaturant, so it will break all the hydrogen bonds. So when we treat this particular protein with urea, so it will be denatured and it will be in the primary state, so it will get opened up. So here the hydrodynamic size would be greater than the initial state of the protein. Okay, so now when we separate this particular group of this will be two groups. What first will be non-denatured, and the second one will be denatured protein because it has been acted upon by urine. So now, if we separate both of them using the gel filtration chromatography, so based upon this hydrodynamic area, this will not be able to enter the pores and it will be eluted first. Okay, so because it will not be able to enter the pores, it will not be able to will not have the sieving effect, so that it will not be able to separate it out. So this will act according to the large molecular weight proteins. Okay, now when the hydrodynamic area is less, so that means this particular protein is fully folded and this will enter the pores, and as this will enter the pores, it will travel across the full column length and it will elute after the denatured protein. Okay, so let's see the options. First option is urea treated protein X will elute So the first option is urea treated protein X will elute after the untreated protein X. So no, the urea treated protein X will elute first as compared to the untreated protein X. So the first option is false. Then we have urea treated protein X and untreated protein X will elute at the same time. No, this will also be not the correct option because of the difference in hydrodynamic area of both the proteins. Then we have urea treated protein X will elute before the untreated protein X. So yes, this is the correct answer. And the last option is peak of urea treated protein X will be better resolved than the peak of the untreated protein X. This cannot be determined on this basis. There are various other factors that depends or that affects the peak of both the proteins. Okay, so the correct answer is option number 3. So any doubts in this question? Okay, so like consider this as a fully folded protein. Okay, so in case of a fully folded protein, it will have a concerted structure. Okay, it will be, for example, it will be a sphere. So this is a fully folded protein. Now if we treat it with urea, it will be denatured. Okay, so it will open up and it will be a string of amino acids, that is a primary structure of the protein. So you consider it as a cylinder. Okay, because now this has opened up to the straight line of amino acids. Now if you will run it through a gel filtration column, for example, these are the pore size or the beads that we have. Okay, so this is the pores and this is the void volume. Now if you see, like this particular sphere, when we add it to the column, it can easily enter the pores and it will flow through the pores and will get separated. But this denatured protein will not be able to enter the pores because here now its area is more as compared to this particular folded protein. Okay, so now if we add it through the column, it will not be able to enter the pores and it will flow through the void volume. Okay, size of both the proteins is same. It is not different. It's only the area or the volume of that particular protein that we are talking about here. Okay, so molecular weight is same in both the cases. Molecular weight is not different. So when this protein is denatured, in that case, it will not be able to enter the pores and it will flow through the voids and it will be eluted first. So this is the denatured protein or the protein that is acted upon by urea, and then the protein that is fully folded, that will travel through the pores and it will elute it afterwards. Okay, so now if we consider the analogy, the denatured protein will now act as a protein that have higher molecular weight. Okay, it although its molecular weight is same as compared to the folded protein, but it will act as a protein that have high molecular weight because it is eluted first based upon its volume. Okay, so now it's understood. Okay, so let's move on to the next question. When a protein is subjected to very high concentration of denaturant, that is greater than 8 molar urea, which of the following is true? It mostly appear in pore volume, it mostly appear in void volume, it will get adsorbed on the beads, or it will elute after native protein. So what can be the correct answer? Okay, any more answers? So as we just discussed now, if we are treating the protein that we have a folded state, so there are different folded state of a protein. Okay, so if we are treating with different concentrations of urea, then we will get different folded states of the protein, okay, based upon the denaturating action of urea. So if we are treating it with greater than 8 molar urea, that means most of the protein will get unfolded. Okay, so if most of the protein is unfolded, then it will appear in the void volume. It will not be able to enter the pores because of its volume. Okay, so now, yes, the correct answer is the most of the proteins will appear in the void volume. So, any doubts in this question? Okay, if not, then we'll move on to the next question. A protein is incubated with a suitable ligand, and the resultant sample was analyzed using gel filtration chromatography. The chromatogram resulted in a single peak. What can be the correct reason for that? All the ligands were bound to the protein, protein was denatured, protein precipitated in the column, or protein was adsorbed on the beads. So, what can be the correct answer? Okay, any more answers? So, here in the protein, we have, here in the question, we have one protein that basically binds to a ligand. Okay, so ligand is a small molecule, and it would have some particular absorbance that we are measuring. So if we run it through a gel filtration column, we would get, we got only one peak. Okay, here the place of the peak is not mentioned, so we'll not get into that particular thing. So we got one peak. So that means what can be the case? The protein is fully bound to the ligand. Then only one peak will be observed. If the protein is partially bound to the ligand, so in that case we would observe three peaks. One that is bound to the ligand, the original protein, and the ligand. Okay, so this will be protein plus ligand. This will be only protein, and this will be ligand only. Okay, so here in the question itself, it is mentioned that it is only observed as one peak. So in that case, it is the protein that has been bound fully with the ligand. So option 1 is correct. Okay, so if the protein is partially bound, then we would observe three peaks. One that is with protein plus ligand, then the other is protein, and the other one, the last one, is the only ligand. Okay, so any doubts? Okay, I have one more question here. If the protein is denatured, in this case, how many peaks would we observe? Okay, so like here we have not mentioned anything about the subunits, so leave it aside. Considering this, yeah, so only, like, for example, if it's a monomeric protein, so we would observe only two peaks, one of the protein and one of the ligand. Okay, and in this case, if the protein is precipitated in the column or protein was adsorbed on the beads, how many peaks would we observe? Yes, and that would be only of the ligand. Okay, there will be no peak of the proteins in this case. Okay, so any doubts in this question? Okay, so let's move on to the next question. A denatured protein has lower retention time as compared to its native state due to uh, this. I have just discussed two questions back, so I think you guys can answer this easily. Yes, the correct answer is option number 3. That is increase in the hydrodynamic surface area of the protein because of the denaturation state. Okay, increase in gel filtration column length would result in reduction in number of theoretical plates, better resolution of peaks, lower column capacity, or resolution remains same as KD is unchanged. What can be the correct answer? Okay, any more answers? Okay, so what happens when we study chromatography? We have a distribution coefficient. Okay, this is related to the analyte. This has no relation to resolution. Okay, so here we are talking about the resolution, and in case of gel filtration chromatography, if we have a column length that is more, okay, or like increased column length, so in that case resolution will be more. Okay, for example, if we have three molecular weight proteins, one is 50 kilodalton, 70 kilodalton, and 100 kilodalton. Okay, so in this case we are using a pore size that can cover these two proteins, and this is left out. So this will be eluted first. This will be the 100 kilodalton protein. Then we have to separate both of them, and we have a similar kind of uh, pore size. Okay, so if in first case we have a column length L1 that is smaller, so in that case the peaks of both the proteins would be not much separated, because the molecular weight difference in both the cases is not very much. Okay, so the difference is very less. Okay, now if we increase the length to L2, that is greater than L1, so in that case what will happen, our peak will change to this category. So here the peaks are relatively well resolved as compared to the initial case, because here the proteins are allowed for longer distance to run on a gel or on a column. So first the 70 kilodalton protein would come out, and then the 50 kilodalton. Okay, so this is the major reason. So here we will be having better resolution of the peaks if we increase the length of the column. Okay, so is the explanation clear? Okay, so let's move on to the next question. A nuclear protein is involved in regulation of gene X. The protein was incubated with gene X and run in a gel filtration column and the chromatogram recorded. Select the correct statement from the following. The protein ligand complex would have lesser retention time. The Kd value of the protein ligand complex will be more. The peak corresponding to protein ligand complex would show a right shift, or the hydrodynamic surface area of the protein ligand complex is less. What can be the correct answer? Okay, any more answers? So this has the similar explanation as we have discussed few questions back. So here we have one protein that basically interacts with the DNA. Okay, so here DNA is the ligand. So their ligand that we used was a small molecule. Here the ligand is DNA. So whenever we make a protein interact with DNA, it would have molecular weight that would be greater than only protein or DNA. Okay, so whenever we'll separate all of them, then this would have lower retention time. That means it will be eluted first, as compared to either only protein or only DNA. Okay, so here it will be eluted first. So the correct answer is option number one. Okay, so the Kd value of this particular complex would be less. Okay, as compared to only protein or only DNA. Okay, so any doubts in this question? If not, then we'll move on to the next question. So we have three proteins that have molecular weights 180, 50, and 250. The proteins with the lowest Kd value is, more answers? So, as per the relation, Kd is inversely proportional to molecular weight. So, which one have the highest molecular weight will have the lowest Kd. Okay, so in these cases, the highest molecular weight is 250 kilodalton. So, it will have the lowest Kd. So, the correct answer is option number 3. Okay, so let's move on to the next question. If a protein is interacting strongly with the stationary phase of a gel filtration column, so in that case, what is the correct relation? Okay, any more answers? So the correct answer is option number 2. So how? Because in this case, if it is strongly attracting or interacting with the stationary phase, so in that case, Bd will be greater than 1. Okay, so if we see the formula, Ve is equal to V naught plus Kd Vi. Okay, so we have the total volume as, okay, so here if we calculate like this, so in this case Ve will be greater than Vt. Okay, so the correct answer is option number 2. Okay, so let's move on to the next question. If the length of the gel filtration column is reduced by half, the Kd of a particular analyte would be. Yes, because the correct answer would be fourth, that will remain unchanged because there is no effect of length of the column on Kd. Okay, so the correct answer is option number 4. Okay, let's move on to the next question. A protein X from Mycobacterium tuberculosis strain is giving a single peak in Cephadix S200 HR10 by 30 column with a retention volume of 12.63 ml. The protein is treated with an increasing concentration of urea and the retention volume will be, so what can be the correct answer in the particular question. Okay, any more answers? So what will happen if we have a protein, we have treated it with urea, it is denatured. Okay. So now if we are separating it using a gel filtration column, the original protein gives a retention volume at this particular ml. Okay, 12.63 ml. So in this case, it is proportional to the retention time. Okay, so when it is denatured, that means it has less retention time. So the volume that we are getting will be less than 12.63. Okay, so now the elution volume would be less than 12.63. So the correct answer is option number 3. Okay, because here the protein is already being denatured, its hydrodynamic area changes, and in that case it will be eluted first, because it will not be able to enter the pores, so it will be traveling through the voids. So that is why the retention value of this particular protein that is denatured using urea is less than 12.63 ml. Okay, so this is the similar explanation that I've been giving for some questions. So let's move on to the next question. The proper packing and presence of bubbles within the gel filtration column can be detected by incubating the column at 40 degrees for 30 minutes, dissolving an analyte that is Kd is equal to 1, washing the column with organic solvents, or dissolving the column in a gradient of ammonium sulfate. What can be the correct answer? So in this case, if we have to detect the proper packing as well as if there are any bubbles that are present inside the column, what will we do? We'll run the column in such a way that the analyte that we are using must travel through the pores as well as through the void jet also. Okay, so in that case, we must have the total volume like this. Okay, so how do we get this volume? By when we have a kd equal to 1. Okay, so this is basically the derivation of the formula from this. So whenever the void volume and the pore volume, so this will be O, sorry. Okay, when the void volume and the pore volume, the sum of these two volumes is equal to elution volume, then in that case, the column that we are using is properly packed and do not have any bubbles. Okay, so in that case, we'll be using a KD that is equal to 1. Okay, so the correct answer is option number 2. So, any doubts in this question? So, if not, then we'll move on to the next question. Okay. The void volume of a gel filtration column is 30 ml. A monomeric protein with a known molecular mass of 25 kilodalton elutes at a volume of 45 ml. Okay, so we are given with void volume that is 30 ml, elution volume that is 45 ml, and that have a molecular weight of 25 kilodalton. The protein that you are trying to purify elutes at a volume of 35 ml. So this is 35 ml. Which of the following is a valid conclusion of this particular answer observation? So we have to predict the molecular weight of the protein that we are analyzing. So what can be the correct answer? Okay. Any more answers? So in this case, we have a elution volume of 45 ml for a molecular weight of protein that is 25 kilodalton. Okay, and the protein that we are analyzing or we are purifying is eluting at 35 ml. So that means the protein of our interest has a volume that is less than the elution volume of this particular protein. So in that case, if the elution volume is less, that means it is eluted out first, and this will be eluted out second. So the molecular weight of this particular protein that we are detecting as molecular weight T would be greater than 25 kilodalton. Then only it will elute out first at a elution volume that is less than the elution volume of this particular protein. That is known. Okay. So the correct answer is option number 3. The molecular weight of the protein would be greater than 25 kilodalton. Okay. So any doubts in this question? If not, then we'll move on to the next question. You are studying a DNA binding proteins in a solution. Three proteins are in solution A, B, and C. and the proteins that have been detected have are shown in here with the following properties. So the isoelectric point is shown and the size of the protein is shown, and the binding property to the DNA is also shown. So what can be the correct options out of which the proteins can be separated through gel filtration chromatography? So here your knowledge of ion exchange chromatography will also be tested side by side. Okay, any more answers? This can be a multiple select answer. Multiple answers can also be correct in this case. Okay, any more answers? So here, in this type of question, first we will see only two columns here: the size of the protein as well as whether it binds to the DNA or not. Okay. So protein A has the maximum size, that is 82 kilodalton. Okay. Then comes protein B, that is 21,500. Protein C 23,000 and protein X is 22,000. Now they have protein A, B, and X can bind to the DNA, but C has no like interaction with the DNA as per the question. So if we have to separate protein X with either of these proteins A, B, and C, we can separate it directly from protein A based upon the molecular weight. Okay, because protein X is 22,000 daltons and protein A is 82,000 daltons. So protein A can be easily separated out because it will elute first and then protein X will be eluted. Okay, so this is the first scenario. So the first option is correct. Okay, now comes the second option, whether we can separate protein X with protein B. So the difference in both the proteins is nearly 500 daltons. Okay, so to get a good resolution in both the cases, we would require a very long column to run with. Okay, that is might be possible, but in this case, if we have another property, so we can use that also to separate out both the proteins, protein X and protein B. Okay, now if we have to separate protein X from protein C, we can use the property that it does not bind to DNA. Okay, because when protein X will be bound to DNA, it would have a elution volume that will be lesser as compared to only protein X or only protein C. Okay, so this will be separated on the basis of these regions. So option number 3 will also be correct. Okay, so we can separate out protein X and protein A and C on this basis. Okay, so the correct answer is option number 1 basically, and option number 3 on the basis of their binding capacity. Okay, so if this type of question comes, then sure shot option 1 would be correct, and if there is a multiple selective question, then option 3 can also be correct, but option 2 is surely wrong. In this case, if we have to separate protein X from protein B because of their similar molecular weight, we'll not be able to separate them on the basis of their size, but we'll be able to separate them on the basis of their isoelectric point. Okay, so we'll be using in that case an exchange chromatography or chromatofocusing. Okay, so is the explanation clear, or there are any doubts in this question? If not, then we'll move on to the next question. So there is a mixture of proteins that are 5 in number and their molecular weights is mentioned. Were separated using Cephadix G50 column. The order of elution of these proteins from the column would be, what can be the correct answer? Okay, option number 2. Okay, any more answers? Okay, so to solve this question, first we would know what is the capacity of Cephadix G50 column. So can anyone tell what is the capacity of G50 column? Please see and tell me the capacity of the G50 column that we are using. What is the threshold that it will separate out? So its capacity is between 1500 Dalton to 30,000 Dalton. Okay, so if there is any protein that is greater than 30,000 Dalton, will travel in void volume as it is. So there will be no separation between those two proteins. Okay, so any protein that is within this range will be traveling through ports. Okay, so now when we'll see the molecular weight of different proteins, so this means greater than 30,000 Daltons is 65 kilodalton and 100 kilodalton. So this means that these two proteins will be eluted out first because they'll not enter the pores at all. So they'll not be able to separate between the 65 and 100 kilodalton. Okay, so they will be separated out at a single peak in void volume. So first D and E will be separated out, followed by the largest molecular weight in this range is 25 kilodalton. So then comes C, then B, and then A. Okay. So the correct answer is option number 4. So are there any doubts in this question? So in this type of question, we must remember what is the threshold or the capacity of different columns if it is mentioned specifically. So here as the G50 column was mentioned, so we have to remember the capacity of this particular column and how now based on the capacity how the proteins will get segregated. Okay, so if no doubts, then we'll finish off the session for today. And you guys have, if you guys have any doubts related to the this week's discussion, you can ask. Okay.